Hello, and welcome back to Learning Lab Diagnosis under 5 minutes. This is part 4 of respiratory infections, and today we are going to learn the most important topic of this unit in any of the exams, that is tuberculosis. So, come, let's learn. Now, this, as you know, is caused by bacteria. So, like any other bacteria that we have previously covered, there are major 7 headings. So, let us learn the bold headings first. First of all, you will collect the specimen, concentrate the specimen, view the specimen under the microscope. These are the 3 headings. Then you will also do the culture, right? So conventional culture, automated culture. Now that you've done culture, you will do culture identification. So these 3 headings, and lastly you will do molecular method. So what have we remembered? We have remembered that we are collecting the specimen, concentrating the specimen and viewing it under the microscope. We are doing 2 types of culture, conventional and automated, and doing the identification, and lastly molecular methods. Now let us start this with our mnemonic sentence. So I want you to remember this mnemonic sentence as a short story. Remember, a morning sputum sample helped Dr. Petrov confirm glowing magic tubes. And so this glowing magic tubes description refers to TB bacteria, alright? In 2 hours, so now Dr. Petrov is taking 2 hours versus Dr. LJ, who is doing it in 6 to 8 weeks. So who's the winner? Dr. Petrov, right? So what do I want you to remember? A morning sputum helped Dr. Petrov confirm glowing magic tubes in 2 hours versus LJ in 6 to 8 weeks. Now let us relate this entire sentence and never forget the broad headings of the lab diagnosis. So in specimen collection, now you might be knowing TB is majorly of 2 types, pulmonary manifestations and extrapulmonary manifestations. So in pulmonary manifestations, as you know, you will be taking 2 types of samples, sputum and gastric aspirate in case of children. Sputum will have 2 specimens, spot and early morning. So now from the mnemonic, you remember sputum, you remember morning, all right, and you can also know that in TB there is pulmonary and extrapulmonary manifestation, so the specimen will vary. Now when you are concentrating the specimen, you are using modified Petrov's method. And now what is this Dr. Petrov using? He is using NALC and NaOH. Hence, you remember N-acetyl L-cysteine plus 2% NaOH. Remember, this number 2 is very common in many parts of this lab diagnosis, so keep a check. Now we are viewing this specimen under the microscope. For the microscope, how will our bacteria appear? It will appear long, slender, it is acid fast. We will also can use cold acid fast staining method, and the bacteria were glowing, right? So we'll do fluorescence or mean staining. So up till now, what have we learned? We have learned that we have learned about the specimen, concentration and the direct microscopy. Now let us go to the culture media. So for conventional culture media, now comes our Dr. LJ. And how is our Dr. LJ? He is a rough, tough and a buff man who will take 6 to 8 weeks to do the same. In automated culture methods, remember MGIT from magic tubes, and you are using that system, which is MGIT system. So this will detect growth and resistance to the anti-tubercular drugs. In culture identification, as you have done in many cases, Malditoff and ICT. In ICT, what will you remember? MPT64 antigen. So we have remembered 2 numbers as of now. First of all, the number 2, which is repeating, and now another number is 64. Okay. Then for molecular methods, you can remember that there is a PCR detective who is looking for IS6110 gene. That will make it easier for you. Then CBNAT, and CBNAT will have a turnaround time of 2 hours. So the relevance of 2 hours here, and line probe assay. Okay, line probe assay is also used for the detecting the resistance. So now come, let's revise. Okay, so for specimen collection, what are we doing? Let us revise as mnemonic first. A morning sputum helped Dr. Petrov confirm glowing magic tubes, that is TB, in 2 hours versus Dr. LJ in 6 to 8 weeks. So for specimen collection, we are taking sputum, which is 2 specimens, spot and early morning. Then in case of children, we are doing gastric aspirate, and in extrapulmonary TB, it will vary on the site involved. For digestion, decontamination and concentration of the specimen, we are doing modified Petrov's method. What is he using? NALC talc and 2% NaOH. For direct microscopy, how do the bacteria appear? Long, slender, acid fast. This is by Ziehl-Neelsen technique, cold acid fast, and we are also doing fluorescent or mean staining method. This are the three headings. In the next three headings, for conventional culture, we will do LJ medium, and it will show rough, tough, buff colonies, and will give us the result in 6 to 8 weeks. In automated culture, we are doing MGIT system, and in culture identification, the common ones, Malditov and ICT, which will detect PT64 antigen. In molecular method, PCR detective will detect IS6110 gene, CBNAT, and line probe assay. So this is the basic outline. Please read this lab diagnosis thoroughly from the book Apurva Shastri, and remember the details so as to fetch full marks, and remember this video and go over it to remember the skeleton and the main points. Thank you. Follow for more.